All right, you are welcome once again to our third lecture in our structures and functions of biomolecules. All right, so today we are going to discuss a subtopic in biochemistry called stereobiochemistry. All right, so stereobiochemistry, when you say stereobiochemistry, is how our biological systems in our body react. Okay, so how they react to molecules when we take them inside our body. For example, for when we take drugs inside our body, how this system reacts to those drugs and how the enzymes in our body react to those drugs. Okay, so it deals with specificity. All right, so they are all specific in nature due to stereochemistry. Okay, so we are going to discuss stereobiochemistry. Okay, and when we say stereobiochemistry, is just a branch of chemistry that deals with stereoisomers, all right, conformation and all those stuff. So we are going to talk about them, and after that we'll talk about chiral molecules. Okay, so we are going to talk about chiral synthesis. So what are chiral synthesis? And we will identify all the chiral synthesis and the chiral carbons, and we give examples so that in case you meet some in the exams or in any test, you'll be able to identify them. And after that, we talk about specific rotation. Okay, so we are going to talk about observed angle of rotation, the concentration and the length, how they are able to make rotate a plane polarized light. So we are going to talk about specific rotation as well, and we will do some basics calculation inside so that you will be familiar with such calculations. And after that, we will talk about enantiomers. All right, we will talk about enantiomers, diastereomers, and meso-compounds. So we will talk about what they are and we'll give examples and all those stuff. And after that, we'll talk about stereobiochemistry. So we will discuss how these biological systems respond to active enantiomers. Okay, so as I said earlier, all the enzymes in our body are stereospecific. So they are optically active and they react to a specific enantiomer of maybe drugs so that it can effectively cure any disease that's intended in the body. All right, so without wasting my time, let's start with stereochemistry. So when we say stereochemistry, okay, so stereochemistry, it is a branch of chemistry concerned with the three-dimensional arrangement of atoms and molecules and the effects of this chemical reaction. Okay, so when we talk about stereochemistry, it just deals with stereoisomers, and they represent molecules in a three-dimensional view. Okay, so all these molecules over here, they are represented in 3D views. All right, so three-dimensional views. Okay, so that's stereochemistry. And we say stereoisomers, they have identical molecular formula. So they are atoms or molecules with the same formula, okay, with atoms bonded in the same sequence, but they differ in the way they are arranged in space. All right, so when we say stereoisomers, so this is the branch of stereochemistry, that's stereoisomers. All right, so within these stereoisomers, we have configurational isomers and we have conformational isomers. All right, so they have the same molecular formula. All right, but their arrangement in space differ. Okay, so let's take example. We have this figure here, we have this figure here. They all have the same molecular formula, but see, the arrangements in space differ from each other. Okay, so that's basically what's meant by stereoisomers. So let's talk into details of these stereoisomers. So stereoisomers is a branch of chemistry that deals with 3D views of molecules. Okay, so under stereoisomers we have configurational isomers and we have conformational isomers. Okay, so these conformational isomers is just the interconvertibility between molecules around a single bond. Okay, so when we check this molecule, we have only single bond here we have 1, 2, 3, 4. So it's a 4 carbon atom. All right, so 1, 2, 3, 4. So it's a 4 carbon atom, and all the bonds involved are single bonds. Okay, so it's conformational isomers. And when we say configurational isomers, these are around a double bond, and some are mirror images too. Okay, so we under the configurational isomerism we have geometric. So geometric is interconvertibility between double bonds. Okay, so we have isomers, and these isomers there are double bonds involved. Okay, and we have optical under configurational too, and under optical too we have enantiomers and we have diastereomers. Okay, so when we say enantiomers, they are molecules, they are mirror images of each other. All right, so we'll talk about it details in few minutes time. So these are mirror images of each other. So let's draw. When we draw a straight line here, you can see that where the chlorine is the same position as where the chlorine is in this second molecule here. So it's just a mirror images of each other. We talk about chiral centers, and you get to know more about enantiomers. And under the optical too, we have diastereomers. These diastereomers, they are not mirror images of each other, although they have chiral centers, but they are not mirror images, and they differ from each other at just a 1 carbon atom. So one chiral center is just the difference between stereoisomers. Okay, so this is stereochemistry, and that's what stereoisomers are. Okay, so let's talk about chiral molecules. Okay, so when we say chiral molecules, so chiral molecules is just an asymmetric molecule that's optically active. Okay, so let's talk about this asymmetric molecule. When we say something is asymmetric, that means it's not having any plane of division. Okay, so it's aplanar. Okay, so it's not having any plane of division. All right, so for example, when we take this compound, for instance, we have CH3 here, we have chlorine here, we have bromine, we have a hydrogen and 1 carbon atom here. So this carbon atom, when we check all the bonds around, it's not having any similar bonds. Okay, so this hydrogen is different from this methyl group, and this methyl group is different from this chlorine, and this chlorine too is different from this bromine. So it's not having any symmetry. So you can't divide this compound into 2, 3, 4, or any else. All right, so it's asymmetric molecule that are optically active, and they rotate plane polarized light. Okay, so all these here are chiral molecules. This is chiral, this is achiral. Because this is achiral, that means we have a plane of division. Okay, the reason why we have a plane of division here is we have a CH3 here, we have a bromine, we have hydrogen here. So there is hydrogen and hydrogen, so we can divide this in this plane, and we can get hydrogen on one half and another hydrogen on the other half. So it's symmetrical, so it's having a plane of division. Okay, so let's talk about chiral centers. When we say a chiral center, it means a tetrahedral carbon atom attached to 4 different group of atoms. Okay, so a chiral center is a carbon atom. So let's take this compound, for instance. We have a carbon atom here, and this carbon atom is tetrahedral. Because why is it tetrahedral? Because it's surrounded by 4 different group of atoms. So that comes the name tetrahedral. So 4 different group of atoms, and this carbon here is chiral because it is not symmetrical. That means it's asymmetry because we have different group of atoms around this carbon bond. So it's different. So hydrogen is different, this is different, this is different, this is different. So we can represent them by A, B, C, D. And A is different from B, B is different from C, C is different from D, and D is different from A. So this compound can be termed as a chiral molecule, and the carbon here can also be termed as a chiral center or a chiral carbon. Why? Because it has 4 different atoms bonded to the carbon. Okay. Alright, so likewise we have achiral. So achiral does not have 4 different atoms or groups bonded to the carbon. This is because it's having 2 hydrogens, and these hydrogens too are similar, so they can't be different. Okay, so we have another chiral compound here, and when you check this compound, we have a metal group, we have an ethyl group, and we have a propyl group here and a hydrogen. So they are all different together. So we have 4 different groups bonded to the carbon. So it can be termed as a chiral molecule and a chiral carbon too. So when we take this compound too, for instance, it has 3 atoms bonded to the carbon. So we have this bond, this double bond bonded to the oxygen. We have a bond single bonded to methyl group and a single bonded hydrogen. So we have only 3 groups of atoms bonded to this carbon. And from the definition of a chiral center, is a tetrahedral carbon atom, and this is trigonal, so it can't be a chiral center. All right, so this is what chiral molecules are, and when we see chiral center, this is what it means. All right, so you can pause the video and you can go through so that you'll be familiar with the terms. Okay, so we have specific rotation. So when we talk about specific rotation, is the degree of rotation that is recorded in degrees. It's an angle of rotation, and it is recorded in degrees, all right, of the plane of polarization of a ray of monochromatic light. When we say a monochromatic light, it's just a light with only one source, all right, a light with only one direction. It's just from one source, so it's a monochromatic light that passes through a tube of 1 decimeter long, okay, 1 dm, containing the substance in a solution at a concentration of 1 gram per millimeter in a polarimeter. All right, so this whole thing summarizes to give this simple equation. So specific rotation is this is the specific rotation, that's alpha here, and this is the observed angle of rotation. Okay, this is the observed angle of rotation that is represented by alpha naught. You can represent it by alpha naught. And this C here is concentration. And this concentration is recorded in gram per mil. Okay. So in calculations, they can give this to you as in, let's say, kilogram per meter or any conversion. So when you are given either cm cube, gram per cm cube, that means you have to convert it into gram per mil before you can use it in calculating what the specific rotation. If not, it's going to be wrong. Okay, and the L2 is the length of part, the part length, and it's also recorded in dm, that's 1 decimeter. Okay, and the changes 10 cm cube is equal to 1 decimeter. All right, so that's the conversion. And the alpha here is the observed rotation. That's the rotation observed for a specific sample. Okay, so this is optical activity. So that's how we measure optical activity of a solution or any solution. All right, and remember the concentration is in 1 gram per millimeter, so gram per mil, and the length is in 1 decimeter. All right, and the angle is measured in degrees. Okay, so take notes of those measurements and the units as well. Okay, so let's do some simple calculations involved in specific rotation. So a sample with a concentration of 0.3 gram per mil was placed in a cell with a length of 5 centimeter. The resulting rotation of the sodium D line was 1.52 degrees in the positive direction. What is the specific rotation? That is the alpha with sodium lamp, that's D lamp. Okay, so that's how it is represented. So how can we go about this question? So when we are given this question, for instance, what we have to do is to write down the parameters. Okay, so the parameters, we have concentration, that's in 0.3, and the unit is in gram per what ml. Okay, so as we talked earlier, the concentration is supposed to be in gram per ml in our previous video. Okay, so the concentration is in 1 gram per milliliter. All right. So after the concentration, it was placed in 5 centimeter length. And remember too, we talked about the length, and we said it's also recorded in dm, that's decimeter. So you have to convert this centimeter to what? Decimeter before you can use it in the calculation. All right. And the resulting rotation at the sodium D line was positive 1.52 degrees. Okay, so what's the specific rotation? So in solving this, you have to first convert the 5 centimeter to a decimeter, and this is the conversion is 10 centimeter is equal to 1 decimeter. Okay, so if 10 is equal to 1, then 5 will be equal to 0.5. Okay, so we just half of it. Okay, so after that, you can start and substitute it in the equation. All right, so we have the specific rotation here, and it's equal to the observed angle of rotation divided by our concentration times the length. And the observed angle of rotation in the question was 1.52 degrees in the positive direction. Okay, that's divided by 0.3 times 0.5, and it's going to give us plus 10.1. So the specific rotation is 10.1 in the positive direction. So this is how you can do some basic calculation in specific rotation. All right, so you can try more examples on it, and it will really help. Okay, so our next topic is enantiomers and diastereomers. So we are going to talk into details what are enantiomers and what are diastereomers. So let's start with enantiomers. So when we talk about enantiomers, they contain chiral centers. Okay, enantiomers contain chiral centers that are not superimposable, and they are mirror images of each other. Okay, so for you to get an enantiomer, one, we have to get a chiral center, and the molecule too, you have to get a mirror image of it. And the mirror images, they are not supposed to superimpose. Okay, so when you place one on each other, they are not supposed to fit. All right, so let's take this for instance. We have an enantiomer here. So enantiomer, they are stereoisomers that are mirror images of each other. So let's take glucose. All right, so this is the molecular formula for glucose represented in a Fisher projection. So this is a Fisher projection of glucose. All right, and this is the D-glucose and the L-glucose. That's the isomers of glucose with this enantiomer. So we have CHO, that's the aldehyde group, and we have hydroxy group here, hydroxy group. All right, okay, so let's check this compound. For instance, when we draw a straight line here, we can see that OH here is here, we have OH here too, we have hydrogen here, we have OH here, we have OH, the same thing, we have OH here, we have OH here. Okay, so this is a mirror image of this one, and when you place this on this, that means the OH here is going to be here, and it's not superimposable. Okay, that means it's not going to fit. All right, so that's an enantiomer. Okay, that's an enantiomer for you. All right, so let's go back to diastereomers. When we talk about diastereomers, they contain chiral centers. Okay, and they are not superimposable, but they are not mirror images too. So for instance, this molecule here, it has a chiral center, and it's not superimposable, but it's not a mirror image of each other. So it can't be an enantiomer. So that means it's a diastereomer. So for instance, let's check here. We have one OH here, which was supposed to be at where the position of the hydrogen was if it was a mirror image. Okay, so since we have an OH here and we have a hydrogen here, it's not a mirror image. Okay, so it can't be an enantiomer. And one note that you have to take in order to identify a diastereomer was here. So diastereomers, they differ from each other in configuration at only one chiral center. Okay, at only one chiral center. So let's check this compound. We have an aldehyde group here, CHO. We have a hydrogen here. We have OH here. And this carbon that is attached to this is also a different group. So this is a chiral center. Okay, the second carbon is a chiral center. All right. And when we take the D-galactose, for example, we have a metal, the aldehyde group here. We have on the second carbon hydrogen OH, and also the third carbon is different group. So it's the second carbon here is also a chiral center. So they are chiral centers, but the position of the hydrogen and the hydroxyl group differ. So it's not a mirror image. So when you check all the other carbons from carbon 3 to carbon 6, they have the same position. Okay, so we have OH here, OH here, we have OH here, OH here. All right, so these are diastereomers. They differ from each other at only one chiral center. Okay, so that's diastereomers for you. Okay, so when we talk about anomers, they are in carbohydrates. So we'll talk about carbohydrates in our next lecture, and you get to understand what anomers are. They are just interconvertibility between two anomeric forms, the alpha and the beta form. So we'll talk about it in our next video that we are going to do. Okay, so this is also a meso-compound. So let's talk about a meso-compound. Okay, so when we talk about a meso-compound, what's a meso-compound? A meso-compound is a non-optically active. Okay, so the underlying word here is not optically active. Okay, so a meso-compound is a non-optically active member of a set of stereoisomers, at least with two chiral centers. Okay, so which is supposed to be of two optically active centers. That means it's having two chiral centers, at least two chiral centers. And this means that despite containing two or more stereogenic centers or chiral centers, the molecule is not chiral. Stereogenic center is the same as chiral center, so don't be confused. Okay, so a meso-compound contains at least two chiral centers and is not optically active, because when we talk about optically active compound, it's not supposed to have a plane of division. So let's take this compound for instance. We have a chiral center here, we have a chiral center here, as the line shows over here. So this is a chiral center because it's bonded to 4 different group of atoms, and the carbon here is a tetrahedral carbon. This also is a tetrahedral carbon. But what we have to know that when we draw a line here, this is a mirror image of this. Okay, so it's having a plane of division, and when we superimpose this on this, it's going to be exactly as the same thing. So it's not optically active. All right, although it's having a chiral center, it's not optically active. So they can ask you in a multiple choice question that is all chiral molecules optically active? And the answer is no, because this is having chiral centers, although, but it's not optically active. Okay. So this is a meso-compound for you, and it's not optically active because it's having a plane of division, although it's having chiral centers, but it's superimposable on each other. So that's a meso-compound for you. Okay, sharp. Okay, so we are going to talk about our last slide, and we are going to talk about stereobiochemistry. Okay, so as I said in the beginning, biological systems, they only have one enantiomer that they are specific to it. So many compounds in the biological system have only one of the enantiomers, okay, that's optically active. So when we take for instance that D glucose and the L glucose that we were talking of, the system in our body responds to only D glucose. It's a sugar. So when we talk about sugars, our body, okay, responds to the D glucose. Okay, that's the sweetest one. So the body responds to D glucose. So it's specific. Okay, so biomolecules, they are stereospecific, and the enzymes in our body too, they are optically active. So they are also stereospecific. Okay, so they have only one enantiomer that's optically active. And this happens because both the enzymes and the cell surface receptors, which metabolize the activities, are all what? Chiral. So they are all chiral molecules. So they are what? Specific to each other. So they react specifically. So the chiral receptor fits the arrangement of the substituent in only one enantiomer. Its mirror images does not what? Fit properly. Okay, so for instance, when we have a drug, let's say we have a specific drug, and every drug too is optically active, so it's supposed to have a mirror image. So we have a D and an L. Okay, so they are enantiomers, so we are going to have a D enantiomer and L enantiomer, but our body will respond to only one of that enantiomers. Okay, so the body will respond to only one type of enantiomer. So it's going to pick either the D or the L based on the one that's going to effectively cure the disease. Alright, so note, for many drugs, only one of the enantiomer is biologically what? Active. Okay, so although they are optically active, but only one is biologically active, because our body is going to respond to only one of the enantiomers. All right, so for example, we have this disease we call Parkinson's disease, and this disease is a disease of the brain that when you get this Parkinson's disease, you'll be suffering from shivering and all kind of stuff that you can't be able to reason well. So it's a very deadly disease, and for this Parkinson's disease, we have a drug called dopamine. All right, so dopamine is able to cure the Parkinson's disease, and this dopamine we have is an optically active, because it's a drug. It's supposed to be optically active. So we have the L-dopa and we have the D-dopa. Okay, so the L-dopa is moderately effective in the treatment of Parkinson's disease, and dopamine, that's D-dopa, okay, is biologically inactive. So the body does not respond to D-dopa, but rather what? It responds to L-dopa. So that's how it's able to treat Parkinson's disease. Okay, and dopamine responsive dystonia, DRD, by stimulating the production of dopamine in the brain. So only the L-dopa is able to stimulate the dopamine in the brain so that it can effectively treat the Parkinson's disease. So that's stereospecificity for you. So biochemistry, we deals with stereospecificity. So enzymes and biomolecules and everything that's in biochemistry, they are all stereospecific. All right. So that's how come we are able to manufacture drug, and the drugs are able to function well, because our body is going to respond to only one specific enantiomer, and that enantiomer is going to help cure that disease. All right, so that's the end of today's slide. So in summary, today's slide it was about stereobiochemistry, and we talked about stereochemistry. We said they have stereoisomers, and we talked about specific rotation. We talked about enantiomers and diastereomers. We talked about a meso-compound. All right, and finally we talked about stereobiochemistry. All right, so that's the end of the slide. And please don't forget to subscribe, like, comment, and share this video for us. Okay, so if you have any comment, you can just enter into the comment section and type any question, or any suggestion that you have for us, and we will answer you immediately. Okay, so kindly subscribe to this channel. And this stereobiochemistry too is very, very important. All right, yeah, because the body needs specificity in order to function, because in case the body doesn't respond to the L-dopamine, unfortunately it responded to the D-dopa. That means it's not the body is not functioning well, and it's going to bring a misfortune, because it's going to produce something that's different from what the body needs, and that's going to be deadly. All right, so that's the end of the slide. And our next video, we are going to talk about carbohydrates. All right, so that's the next video for our structures and functions of biomolecules. Okay, so stay blessed, and as I said earlier, don't forget to like, comment, and subscribe to this channel for us. All right, thank you.